In this video, we are going to start to talk about the producers of the salt marsh. We are going to go over objectives 12 through 15. And so I'm going to start with objective 12, which talks about a micro producer, something very small. You need a microscope to see diatoms. We are going to talk about diatoms in almost every ecosystem. They're incredibly important for our oxygen production. But this objective goes over the structure of their shell and the chemistry of the shell. Their shell is called a frustole, so let's write that word down. And it's made out of silica, which is basically glass. And the reason why that's important is because their shell is kind of susceptible to ocean acidification and other things that will break down glass. Also, because it's made out of glass, it creates fossils, which is awesome because we can look at past fossil record of diatoms, and actually it will tell us the history of atmospheric temperature. We'll go over that part in class a little bit later, but the structure just looks, it has two pieces to it. It has a top part and a bottom part, and they fit together kind of like a clothing box. And so one thing to note that on the shell is they are covered with microscopic pores. And what do you think the pores are for? Well, I hope you guessed gas exchange because they are photosynthetic, and so carbon dioxide is going to go in and oxygen is going to go out. So let me see if I can actually draw that on here. So you have CO2 coming in and you have oxygen coming out through the process of photosynthesis. Okay, so this is how they reproduce. This is the second part of objective 12. They produce both sexually and asexually, and so we're just going to briefly go over these. This is not something that we are going to stress this unit, but it's nice to know that they can produce both sexually and asexually. And so asexually is how they normally reproduce, but I want you to know that it uses mitosis, but they get smaller with each division. And at some point they become inefficient. If you look over here, these are all different sizes. Some of these smaller ones, they become pretty efficient. So when they become that small size, they actually start to release, they can recognize that, and they start to release their sperm and egg, which will create a cell called an oxospore, which is right here, which will grow large and become a larger diatom that will then repeat the cycle. So asexually is through mitosis, sexually is going to be through fertilization. But it's only until they get to that really small point right here that they become inefficient, and then they have to reproduce sexually. All right, objective 13 says that you can draw the structure of a dinoflagellate and explain their involvement in HABs in an estuary. HAB stands for harmful algal bloom. So if you want to write that down, HAB is a harmful algal bloom. And we first mentioned those words when we watched the TED Talk video on biodiversity. Algal blooms are due to nutrient overloads, and sometimes we're going to learn there are parasitic algae that can bloom and then become a harmful algal bloom. Usually they're red, also called red tides. A dinoflagellate, for our purposes now, are going to be found almost in every ecosystem. They have positive ones, they have negative ones, meaning they have ones that have a symbiotic relationship, they're very positive, like in a coral reef, and then they also have ones that are parasitic, that are linked to harmful algal blooms. How would you identify them? Well, you can look at the pictures over here, and it looks like they have two shells. So I want you to kind of draw a shell here. It's almost like a hamburger bun, if you will, and then draw a flagella. Okay, that is going to be our dinoflagellate. Here is a video on dinoflagellates, and so you can see them moving around. This is from a salt marsh. I'm just going to point out a couple things as the video progresses. If let me pause that real quick. Up here, you can see, let's see if I can write, up here you can see the double shell. Over here and over here, you can't see the flagella. But again, these are microscopic, and they are moving through means of a flagella. Something that we need to write down that I did not write down in the last slide is that they are not made out of glass. Their shell is made out of calcium carbonate. Okay, so I'm going to pause this video, and I'm going to go back to our notes, and we're going to write that their shell is made of calcium carbonate. Again, ocean acidification, the lowering the pH, is going to affect them as well. These are both examples, diatoms and dinoflagellates, of photosynthetic organisms. So when we are doing destruction to their population, we're directly affecting our oxygen levels as well. Keep that in mind. All right, what we're looking at here is a website found by Andrew. So if you go to the outline, underneath Objective 13, you'll see Andrew's link right there. It compares diatoms and dinoflagellates. I like this website a lot. Here's a diagram. Here is your diatom. Here's a dinoflagellate. You can see the flagella right here. What I want you guys to take note of is what I use my Digo for, is some of the differences. So kind of going back to diatoms, they're eukaryotic, they're single cell, they're algae, they're photosynthetic, which I already mentioned. Their shells are made out of glass. I said it looked like a clothing box. They described it as a petri dish. Same thing. And very important in the fossil record. That's something to note because we are going to talk about what information we can get from them. And I told you, they're all over. There's over 100,000 species. They're very beautiful. They come in all different shapes and sizes. So, this is just one look of a diatom. They have many different looks to them. Dinoflagellates, some do have two flagellas. Some of the species I've looked at have had two. We drew ours with one. And they actually can be autotrophic and heterotrophic, so some are capable of photosynthesis, whereas some actually are a little more heterotrophic and will consume other organisms. Not as many species known, only about 2,000, but we can link 60 of them to these toxic, harmful algal blooms we've mentioned before. So if you read here, when conditions are favorable, a population explosion or bloom may occur, sometimes resulting in the contamination of fish and shellfish, and posing a threat to humans and animal health. That is a concern on the Jersey Shore. We'll talk about Pfisteria when we get to class. One area in a salt marsh that we're definitely going to smell is the mud flats. And because the mud is so dense, even the roots of plants have to kind of go through anaerobic respiration to make energy because of the lack of oxygen in the mud. And so because it's so dark and dense, obviously light is also not going to be able to survive down there. But there are bacteria and other species down there that need sugars, that need some type of energy form to even be able to respirate. You need the energy first. And so the processes that trap our energy from the environment are photosynthesis and chemosynthesis. So you might want to write that these processes definitely trap our energy from the environment. So let's write that first. They trap energy from the environment. And then I want you to kind of make a T-chart. Oh, all right, like that. And I want you to make a T-chart that compares and contrasts photosynthesis to chemosynthesis. And I want you to turn to page 98 in your book, on page table 5.1, which I'm going to have on the screen right here. I want you to pause the video, and I want you to kind of go over the differences. All right, so pause the video and look at this chart and compare photosynthesis to chemosynthesis. All right, so you should have opened up your book and had this table. It might be hard to read off the screen because I had to enlarge it. But the table 1.5 in your book goes over photosynthesis, chemosynthesis, and we'll get into the respirations later. But the source of energy is what makes them significantly different. You have photosynthesis, whose source of energy is light from the sun. Okay, whereas you have chemosynthesis, which is a long list of chemicals ranging from sulfur, ammonium, nitrate, and iron. The one that we're going to talk about today is going to be sulfur oxidation. Okay, now something that you might find a little interesting, which you should have, is the other raw materials, the other reactants. When you look at photosynthesis, you guys should know this by heart. Carbon dioxide, water, okay? Making oxygen and glucose. But did you notice this? Chemosynthesis uses oxygen as a reactant. And so that's something that should have been interesting to you. The last column here, production of organic carbon, yes, it's going to make some sort of energy. It may not be glucose, but it's going to be some form of a carbohydrate. And so going back to our chart, I cleaned mine up a little bit. I want you to pause and write this down. Photosynthesis has light energy, whereas chemosynthesis chemical energy. Pause the video and write these chemical equations down. All right, now that you have these down, I want to label the reactants and the products so that we can look at them a little more visually. So over here in photosynthesis, I want you to underline carbon dioxide and water, those are the reactants. And we know it makes glucose and oxygen, which are the products. But in chemosynthesis, it uses carbon dioxide. You need the carbon to make the sugar. This is the chemical that it's using, but also needs oxygen, and that's important. Where does that oxygen come from? These are the reactants, and these are the products. So I want you to circle this oxygen and put a question mark, because in class we need to figure out where does that oxygen come from. This process happens deep in mud flats. For us in the salt marsh, it's going to happen in the bottom of the ocean. Where does that oxygen come from? Our last objective, 15, I can explain why the mud flats in a marsh would benefit from chemosynthesis. So turn to page 265 in your book, and I want you to read the paragraphs that talk about chemosynthetic bacteria. So pause this video and make sure you read page 265. All right, now that you've read the page, I want you to look at the, let's explain why they would benefit from chemosynthesis. First of all, what species is abundant? Look in your notes. Can you answer that? Bacteria are extremely abundant in all soils and especially mud flats. And usually bacteria's job, some are photosynthetic, but most of them is for decomposition. Okay, something you need to know about decomposition is that it uses oxygen. All right, so it's going to use up oxygen. If oxygen is not available, then it can perform anaerobic. And in the mud flat, we had already said it's not going to use any oxygen because oxygen is really not there. So what happens is it's going to go through anaerobic respiration. And a product of anaerobic respiration is hydrogen sulfide. And so hydrogen sulfide, if you remember going back to our table, is one of the reactions. H2S is hydrogen sulfide. So that's actually a reactant of chemosynthesis. Okay, so anaerobic respiration produces H2S, which is hydrogen sulfide. Okay, and so in turn, we know that anaerobic respiration is going to produce a product that becomes a reactant in chemosynthesis. So let's write that down. Anaerobic respiration produces a product that is a reactant in chemosynthesis.